This video is about cancer. Cancer is a collection of diseases with the common feature of uncontrolled growth of cell. Cancer is a term in which cells are no longer fully responsive to signals that regulate cellular differentiation, proliferation, and cell death. Therefore, these cells begin to division fast and unregulated cell growth. These cells form a mass called a tumor. The tumor may be two types: malignant tumor and benign tumor. In malignant tumor, cells divide and grow uncontrollably, which may invade nearby parts of the body. The cancer may also spread to more distant parts of the body without the lymphatic system, through the lymphatic system or bloodstream, known as metastasis. Not all tumors are cancerous. The benign tumors do not invade neighboring tissues and do not spread throughout the body. The abnormalities Not all tumors are cancerous. The benign tumor do not invade neighboring tissues and do not spread throughout the body. The abnormalities in cancer cells usually result from mutations in protein encoding genes that regulate cell division. Now let's learn about genetics of cancer. The genes that involve in cancer can be divided into the following types: proto-oncogenes and tumor suppressor genes. First, proto-oncogenes. Protein encoded by proto-oncogenes may function as growth factors or their receptors, signal transducers, transcription factors, or cell cycle components. Proto-oncogenes regulate the cell growth, division of cell, prevent cell differentiation, and regulate apoptosis. Example of proto-oncogene. One of the well-known example of proto-oncogene is the HER2 gene. The gene encodes for a transmembrane tyrosine kinase receptor called human epidermal growth factor receptor. This protein receptor is involved in the growth, repair, and division of cells in the breast. WNT, MYC4 gene, RTK, RAS-MAP kinase are proto-oncogenes. Products of proto-oncogenes genes are essential for cell survival and growth. Mutations in proto-oncogenes convert proto-oncogenes into constitutively active cellular components or oncogenes that are involved in tumor development. Mutations in proto-oncogenes increase the production of these proteins, thereby leading to unregulated cell division, slow rate of cell differentiation, increased inhibition of cell death. These are the common features of cells that have become cancerous. So, proto-oncogene in DNA. Translocation or transposition, gene moved to new locus under new controls, new promoter. Normal growth stimulating protein in excess. Then gene amplification, multiple copies of a gene. Normal growth stimulating protein in excess. Then point mutation within the gene. Oncogene, hyperactive or degradation resistant protein. These are the conversion ways of conversion of proto-oncogene to oncogene. Like gene amplification, multiple copies of a gene may be produced. Normal growth stimulating protein in excess. Then translocation or transposition, genes move to new locus under new control. So normal growth stimulating protein in excess. Then point mutation within the gene, hyperactive or degradation resistant protein. Then proto-oncogenes convert into oncogenes by the following type of mutation pointing mutation. A proto-oncogene is converted into oncogene by insertions or deletions that give rise overactive gene product. These mutations also lead to the increase in transcription rate. For example, a change in RAS oncogene include HRAS, KRAS, and NRAS. The proto-oncogenes code for signal transduction. A GTP binding protein has TTPase activity that become active to inactive by hydrolysis of GTP to GDP. The RAS protein P21 intrinsic GTPase activity that is regulated by the other proteins. Then gene amplification leading to additional copies of proto-oncogene. Many oncogenes have been identified in amplified regions. The ERB2 is a transmembrane growth factor receptor which has tyrosine kinase activity which involved in cell growth. In rat neuroblastomas, the ERBB homology has a T to A mutation that changes glutamate to valine in the transmembrane region. This facilitates ligand-independent dimerization of the receptor, therefore it is constitutively active. Hence, receptor activity is increased by increased normal protein in human and an abnormal constitutively active product in rat. Then growth factors. Growth factor receptors are receptors tyrosine kinase pathway. Growth factors are required by all cells to undergo proliferation. Growth factors has paracrine action. However, many cancer cells start expressing the growth factor to which they are responsive, generating an autocrine loop. Growth factor receptors can be constitutively activated in tumors by various mechanisms, including mutations, gene rearrangements, and overexpression. So, glioblastomas secrete platelet-derived growth factor (PDGF) and express the PDGF receptor. Sarcomas secrete transforming growth factor TGF alpha and express the receptor. Sometimes the growth factor is not mutated, but a transcription factor like RAS is mutated. Mutated RAS containing cell express large amounts of growth factors such as TGF alpha. Present ovarian cancers, FLT3 receptor, FMS like tyrosine kinase 3 is amplified on cell surface. Then leukemia cancer is due to point mutation in the RET receptor for neurotrophic factors. Then squamous cell sarcoma of lungs, gliomas is due to overexpression of ERB1, EGFR, ERB2. These proteins are EGF receptor. Gastrointestinal stromal tumors, seminomas are because of point mutation in KIT. KIT is a receptor for stem cell factor. RET proto-oncogene receptor tyrosine kinase 4 glial cell line. It promotes cell survival during neural development. RET loss of function mutation are associated with the development of Hirschsprung disease. I'm not sure about the pronunciation. Hirschsprung disease. I'm not sure. While gain of function mutations are associated with the development of various types of human cancer. Then signal transducing proteins. The most well-studied example of signal transducing oncoprotein is the RAS family of guanine triphosphate GTP binding proteins or G proteins. The RAS oncogene, 3 types of RAS genes are associated with cancer in human genome: HRAS, KRAS, NRAS, NRAS and KRAS. RAS oncogene is originally discovered as retroviral oncogene and is constitutively activating RAS. Mutations have been identified in human tumors. The RAS gene, of which there are 3 in the human genome, HRAS, KRAS and NRAS, were discovered initially in transforming retroviruses. The single most common abnormality of proto-oncogene in human tumors is point mutations of RAS family gene. RAS is the most important downstream signaling of growth factors. RAS is a G protein. When RAS is bounded with GDP, it is inactive, and when it is bounded with GTP, it is active. The activated RAS stimulates downstream regulators of proliferation such as the mitogen activated protein MAP kinase cascade. The orderly cycling of the RAS protein depends on 2 reactions: nucleotide exchange DDP by GTP, which activates RAS protein, and GTP hydrolysis, which converts the GTP bound active RAS to the GDP bound inactive form. GEFs and GAPs, critical element in the control of small g protein. So signal G protein GDP like this. So guanine nucleotide exchange factors or GEFs and GTPase activating proteins GAPs regulate the activity of small guanine nucleotide binding protein to control cellular functions. In general, GES turn on signaling by catalyzing the exchange from G protein bound GDP to GTP, whereas GAP terminates signaling by inducing GTP hydrolysis. Several different point mutations of RAS have been identified in cancer cells. The point mutation reduces the GTPase activity of the RAS protein. The cell is forced into a continuously proliferating state. In colon, lung and pancreatic tumors, point mutation is found in KRAS. In bladder and kidney tumor, point mutation is found in HRAS. In melanomas, tumor point mutation is found in NRAS and also in BRAS. In hepatoblastomas, hepatocellular carcinoma tumors, point mutation causes overexpression of B-catenin. B-catenin is involved in WNT signal transduction. In acute lymphoblastic leukemia, AVL is translocated. AVL is a non-receptor tyrosine kinase. All the cancer therapies are targeted toward anti-RNA modalities, but unfortunately, none of these strategies has so far proven to be successful for clinical use. Then transcription factors. Some protein work as transcription factors are involved in tumor formation. The MYC oncogene. CMYC is a regulator gene that codes for a transcription factor. The transcription factor plays a role in cell life progression, apoptosis, and cellular transformation. Overexpression of the MYC protein is commonly found in tumors. In Burkitt lymphoma, a B cell tumor of the MYC gene is translocated to Burkitt lymphoma. A reciprocal translocation between chromosome 14 and 18 is also extremely common to follicular B cell lymphoma. This translocation leads to the fusion of the BCL2 gene with the IgG locus, resulting in anti-apoptotic protein BCL2 overexpression. So, chromosome MYC gene reciprocal translocation between chromosome 14 and 18 is shown in this figure. Dysregulation of MYC expression resulting from translocation of the gene occurs in Burkitt lymphoma, a B cell tumor. MYC is amplified in some cases of breast, colon, lung, and many other carcinomas. The related NMYC and LMYC genes are amplified in neuroblastomas and small cell cancers of lung, respectively. So, alternations of non-receptor tyrosine kinase, expression of MYC gene is shown. CMYC, cell life progression, increase cyclin D1, increase cyclin D2, activate CDK4, activate CDK2, decrease P21, decrease P27, cellular growth and differentiation, suppress GAS1 and suppress GADD45, then cell adhesion, decreased LFA1, downregulates collagen production, decrease fibronectin. Then cellular metabolism stimulates LDHA product, increases glucose transferase 1. Then activate phosphofructokinase. Then apoptosis induces P19, increases P53, activates FAS or FAS ligand. Then unregulates BAX. So alternations in non-receptor tyrosine kinase. Alterations in non-receptor tyrosine kinase. Mutations in non-receptor tyrosine kinase are also associated with cancer. Normally, the type of mutation is chromosomal translocation or rearrangements that create fusion genes encoding constitutively active tyrosine kinase. An important example of this oncogene mechanism involves the chromosomal translocation. The chromosomal translocation causes a proto-oncogene to move to a different chromosomal site associated with increased expression. The translocation also causes a proto-oncogene to fuse another gene to produce a protein that has oncogenic activity. Then retinoic acid receptor, acute promyelolytic leukemia. APL is a leukemia in which differentiation is blocked at the cell stage. This is a reciprocal chromosome translocation. The RARA receptor binds with retinoic acid, forms a functional transcription factor by heterodimerizing with retinoic acid receptor plus its ligand cis retinoic acid. The PML RARA fusion protein will heterodimerize with retinoic acid X receptor and interfere with RXR functions. The RARA gene from chromosome 17 is translocated next to PML gene on chromosome 15. This translocation generates two different PML RARA fusion proteins. Also, loss of part of RARA transactivation domain interfere with trans-retinoic acid function. Example is CABL tyrosine kinase. The ABL1 is a proto-oncogene and encodes a cytoplasmic and nuclear protein tyrosine kinase, which involves cell differentiation, cell division, cell adhesion, and stress response. In CML and ABL gene is translocated from its chromosome 9 to chromosome 22, where it fuses with the RCR gene. Activity of ABL1 protein is negatively regulated by its SH3 domain, and mutation or deletion of its SH3 domain convert ABL1 gene to oncogene. The resultant chimeric gene product is constitutively active. Oncogenic BCR-ABL tyrosine kinase BCR-ABL protein can be inhibited by various inhibitors. So normal chromosome 9, normal chromosome 22, BCR and ABL, chromosome break, BCR and ABL, changed chromosome 9, BCR and ABL can be seen. Then viral carcinogenesis. Carcinogenesis is the mechanisms where viruses cause cancer, that viruses carry out a gene altered the cell regulation. Example of viruses that contribute to carcinogenesis, there are two classes of tumor viruses, RNA viruses and DNA viruses. RNA virus infect competent cells. Their RNA is converted into DNA that incorporated into the host genome. Hence, that is classified as retroviruses. Examples of retrovirus: human T lymphotrophic virus, HTLV1, and Rous sarcoma virus, RSV. RNA virus show two general ways: provision of an oncogene or insertion mutagenesis, in which regulatory sequence alter host gene activity. Then provision of an oncogene. Many oncogenic RNA viruses contain an oncogene additional to the sequence for viral replication. The Rous sarcoma virus, in which the oncogene VSRC code for a 60 kilodalton phosphoprotein that has tyrosine kinase activity. Insertion or mutagenesis. Mouse mammary tumor virus MMTV has regulatory sequences that can stimulate host gene. The regulatory genes are repeated at each end of viral genome. Long terminal repeat. The LTRs contain enhancer sequences capable of increasing transcription from nearby genes that codes for fibroblast growth factor. HTLV code for transcription factor that regulate host genes such as FOS. Then DNA viruses. Several DNA viruses are specific in human cancers: human papilloma virus, hepatitis B virus, and Epstein-Barr virus, Kaposi sarcoma virus. The cellular response of these viruses derived from the protein E6 and E7. The E6 and E7 mRNAs are generated by cleavage of larger RNA. Carcinogenesis is time-dependent, multi-stage process. Inactivation of RB and P53 by viral E6 and E7 proteins. This progress of disease is favored by various cofactors such as tobacco, smoke, and oral contraceptives. If a proto-oncogene undergoes a somatic mutation, control of cell growth is lost in the cell in which the mutation occurs, and cancer can occur. This proto-oncogene, which have been shown to mutate in any individual, is called cellular oncogenes and are designated by the prefix C, CMYC, cellular oncogene, to distinguish them from the viral oncogene. These proto-oncogenes that have not been found to mutate are called normal oncogenes and are designated N. Next, retrovirus associated oncogenes. The oncogene of those virus transforming a cell to unproliferated growth carry an oncogene in addition to three primary genes required for all retroviruses. The figure shows the generalized structure of a retrovirus: LTR, GAG, POL, ENV, UNC, and LTR. Long terminal repeat: capsid protein, reverse transcriptase, protein spikes, oncogene, and long terminal repeat. So this is an example of retrovirus. Rous sarcoma virus. The oncogene found in this retrovirus is SRC. SRC is a receptor tyrosine kinase and involved in cell division and cell proliferation. After the infection of Rous sarcoma virus, the cells start expressing more SRC, which leads to tumor formation. The avian erythroblastosis virus. The oncogene found in this virus is VERB. The ERB is epidermal growth factor receptor. VERB is a receptor tyrosine kinase and involved in cell division and cell proliferation. After the infection of avian erythroblastosis virus, the cell start expressing more VERB receptor on cell surface, which leads to tumor formation. Then Harvey murine sarcoma. The oncogene found in this virus is VHR-RAS. VHRAS binds to GTP. VHRAS involved the cell division and cell proliferation. After the infection of Harvey murine virus, the cells start expressing more VHR-RAS, which leads to tumor formation. Tumor suppressor genes. Tumor suppressor genes. Earlier we have learned about proto-oncogenes. Next one, tumor suppressor genes. Tumor suppressor genes are genes that slow down cell division, involve DNA repair and apoptosis. When tumor suppressor genes don't work properly, cells can grow out of control, which can lead to cancer that is created by loss of function mutation. In contrast, active mutation inactivated oncogene from proto-oncogenes. Tumor suppressor genes are proteins that are encoded. Tumor suppressor genes suppress the tumor. Tumor suppressor gene control the processing of genetic of integration and the procession of cell cycle, differentiation, cell interactions, and apoptosis. Tumor suppressor genes are inactivated by mutations. Tumor suppressor genes are inactivated by mutations, contribute to the loss of tissue homeostasis, the hallmark of developing neoplasm. The open reading frames of tumor suppressor genes are commonly truncated by nonsense mutations, frame shifting mutations, small addition or deletions, or splice site mutations. Sometimes exon removed by larger deletions. Tumor suppressor gene also inactivated by alteration of single nucleotide residues in tumor suppressor proteins. There are encoded protein become non-functional. TSG can be grouped into categories including caretaker genes, gatekeeper gene, and transcaper genes. Retinoblastoma. It is caused by loss of both alleles of a gene. RB is tumor suppressor protein. Mutation in RB results in cancer. Normal cell express suppressor protein that inhibits the cancer. Retinoblastoma. RB is a large gene. Although mostly mutations are in 30 kb coding region and mostly involve chromosomal change, several retinoblastoma can arise in one eye, each with a different RB mutation. The RB protein has about 10 phosphorylation sites. RB has an ability to interact with other proteins. Over 25 RB finding protein has been identified with functions such as nucleosome structure, tyrosine phosphorylation, oncogene, and transcription factors are responsible for proliferation. The RB protein has more than 10 phosphorylation sites. Conversion from hypo to hyperphosphorylated sites changes the interaction of RB with other proteins. RB binds with transcription factors E2F. It is regulated by phosphorylation. The hypophosphorylated state of RB, the E2F is bound. This makes E2F inactive. The proliferation of cell cycle is regulated by cycling dependent kinases or CDKs. The cycling dependent kinases phosphorylate and inactivate RB, thereby relieving the cycle block. The released E2F stimulates the transcription of genes that regulate growth such as CDC2, MYC, and DNA polymerase. RB also inhibits transcription from rRNA and tRNA genes by binding of UBF, upstream binding factor, and TB3B, transcription factor 3B. RB virus influence the mass of a cell, protein content, and RB repression occurs normally by hyperphosphorylation and abnormality by RB mutation by binding other proteins. The p53 tumor repressor gene is activated in response to a wide variety of cellular stresses including DNA damage, ribonucleotide depletion, redox modulation, hypoxia, changes in cell adhesion, and stresses created by activated oncogenes. The p53 protein work as transcription factor and activate genes that are involved in DNA repair, apoptosis, and growth arrest. These activities of p53 help in maintaining the genomic stability, hence p53 is called the guardian of genome. If normal p53 is mutated and non-functional by binding of other proteins, p53 has 4 functional domains involved: transcription activation domain, TAD, DNA binding domain, oligomerization domain, autoinhibitory domain. Each domain binds to several proteins that regulate p53 function. p53 binds to its response element present upstream to the gene to transcribe the gene. p53 binds in tetrameric form. p53 increase the expression of BAX, p21 insulin-like growth factor binding protein 3, GAD45, and thrombospodin. Expression of genes such as BCL2, FOS, and GIN can be inhibited by p53. Thus, p53 inhibits cell proliferation. The gene p21 that code for cycling dependent kinase inhibitor p21 inactivates CDK that is essential for DNA synthesis. GADD binds to protein proliferating cell nuclear antigen that needed for both DNA synthesis and repair. Hence, p53 inhibits DNA synthesis while allowing repair to continue. DNA damage activates p53 function by post-transcriptional and cell type specific mechanisms. Normal p53 show inactivation by protein binding. For example, adenovirus codes for E18 that binds to TAD of normal p53 and block its transcription. Some human sarcomas have MDM2 gene that have some end results. Human papillomavirus have protein E6 that binds to OD and prevents dimerization, while the HPX protein of hepatitis B virus binds and inactivates P53. P53 transcribe the thrombospodinin PA1, KA1, BA1, and they block the angiogenesis mutation in P53 thrombospodin like that. Damage to DNA, general regulators, MDM2, etc., apoptotic specific regulators, P53 increased, P21CIPI, DNA synthesis inhibited, then DNA repair activates, then apoptosis stimulated.